Hi there, AP Bio students. So I thought I would go through some FRQ examples with you and kind of talk you through them. And I will post this PowerPoint right now. One of the questions is completely thoroughly done. The rest of the questions have the answers in there, but not my discussion comments. So as you go through this PowerPoint, it's called Sample FRQ Questions with Weith. This first one here from the AP Bio course description, question number 1, totally worked out. You can see the question here, here, here. It's a long one with a lot of content. There's 3 visual aids. And then there's the questions, A, B, C, D for 10 points. And then there's a page or a slide that says wait, before you move on, maybe try it yourself. And then it gets followed by these green slides with the answers. Then you'll see this next teal, gray page. That one's done. This is the next one from the AP Bio practice exam number 1 for this year. I do 1 and 4. Then from number 2, I'll do number 1, and then I'll put in some past student work. So for right now, this question from the AP Bio course exam description, question number 1, is the only one that I'm going to talk about in this video. I'll still post the PowerPoint as it is for right now. I'll post the video as well, and then as I update deeper into the PowerPoint, I'll update the PowerPoint and then add another video link for the next question, So this won't be an endless video feed or endless PowerPoint. So you will have 2 questions on your AP Bio test. Style number 1 is interpreting and evaluating experimental results. They say it's 8 to 10 points, but for this year, you're not going to know how many points it's actually worth. But part A is going to actually have you describe something. Oh, I can't. Oh, well, fine. Describe and explain what's going on in the model, so you're applying content knowledge to this. Identifying experimental design things. Independent variables, dependent variables. What is the null hypothesis that this experiment should have? Why are they doing a negative control or a positive control, which I'll talk about. And then analyzing data, you look at the data that's provided from the experiment and you do something with it. And then usually they take one step further, now the researchers want to do this, make a prediction. Or now the researchers have this claim based on new data, what evidence supports that claim. So that's pretty common to question style 1. Question style 4 is shorter. It's usually around 4 points. And now you have a scenario and there's a disruption to the biological scenario. So you have to, look at the scenario as is and apply the concepts that you know about. Explain what's going on. So you're maybe describing an element, just straight up content, explaining what's going on in the scenario, and then predict what's going to happen with that change, and then justify your predictions with more content understanding. So similar to the first one, but not related to a lab. It's probably more like a case of biology that you are used to. So let's see, the first one. So in many countries, these mosquitoes, I'm going to read it to you as if I were reading it internally to myself. I wonder, I don't think I can mark up the screen, can I? Yeah, I think I can. Okay, great. So I'll do some screen marking up in a moment. But I'll kind of read you through and then go through A, B, C, D, and hopefully I'll make it in the next 10 minutes. So, mosquitoes are responsible for transmitting the parasite that causes malaria to people through their bites. So it's kind of like a vector. So the parasite is infectious to people, it gets in the mosquito puts it in the person. A primary tool for mosquito control is the use of insecticidal nets sprayed with chemicals known as pyrethroids, I'm just going to call them P's, which are relatively safe for people but toxic to mosquitoes. However, mosquito resistant to P's has now become widespread, so this insecticide is not working. P's interfere with the function of a transmembrane sodium channel found in cells of the mosquito. So here's the membrane, and then this is your protein. Transmembrane meaning it goes through the membrane, on this side, through the membrane, on this side, through the membrane, through the membrane. Here's the amino end of the amino acid, the carboxyl end, and regions 1, 2, 3, and 4. And the P insecticide interferes with the sodium channel, and that's how it's used to kill mosquitoes. So, in one common mutation to the protein, phenylalanine is substituted for a leucine at position 1014. Scientists hypothesize that this mutation is responsible for some cases of this insecticidal resistance. So, oh boy, sleepy today. The insecticide normally works if the mosquito has a leucine at that site, but if some of them get the mutation have phenylalanine, now they have resistance. Okay, so to investigate P insecticide resistance, mosquitoes were collected 4 times over a 2-year period for the following 2 regions. So I'm going to go to the next slide. So we have region A, vegetable growing region where large amounts of insecticide are applied. So region A has lots of them. It looks like we have 2 chemicals, different times, and percent mortality. So lots of mortality with this one, less mortality with that one. Region B, rice growing region, very little insecticide is applied for the rice. And if you look at the time, 2008, 9, 2009, 2010, so we had lots of death of mosquitoes and then less. Lots of death of mosquitoes and then less in region B. Scientists exposed the collected mosquitoes to filter papers soaked with 2 different P insecticides. I can't pronounce, and this one. And the percent mortality of mosquitoes would determine after 24 hours. So that's what's going on in this diagram. The scientists simultaneously determined whether leucine or phenolphthalein was encoded by each of the 2 copies of the sodium channel gene. So now you have this chart, and so you have region A mosquitoes from the different times. So notice it goes opposite of the graph. The graph is starting with far back in the past at the bottom, going up to more current times, 2010, where it starts with in the past and then goes down to 2010. So just be careful with that. Total mosquitoes tested, and then are they homozygous for having leucine at that site? Notice they would have like 2 chromosomes, and what letter on the DNA on this chromosome would end up coding for the gene, right? And then what on this one? So they got 2 copies. Homozygous for leucine would be 2 leucine ones. That's the one that is the original, right, that would let the mosquitoes be killed. Heterozygote for leucine and phenylalanine, they got 1 of each allele. And then homozygous for phenolphthalein, they are able to be resistant and they're making the phenylalanine. So you can see a lot more of those at region A. So there's a lot less mosquitoes dying in region A. What? Figure 2. Susceptibility of these mosquitoes from 2 regions to these insecticides, D and this one. A mosquito strain that is susceptible to the insecticide displayed at least 95% mortality in all experiments. So they're testing a mosquito strain that definitely dies from this insecticide, and it kills 95% of them. That's a way of like checking that that insecticide works. And mosquitoes exposed to untreated filter paper displayed less than 10% mortality. So if a mosquito is not exposed to an insecticide, they expect to see about 10% of the mosquitoes die. So that's another kind of control they're doing. Error bars represent standard deviation. ND means no data is available. So, all right. Nothing. Oh, nope, that's the same thing. Okay, so the questions. A. Describe the most likely cause of the amino acid substitution in the sodium channel protein, and explain how the substitution of a single amino acid in the channel protein could cause this insecticidal resistance in mosquitoes. So let me go down here. So this is where you would pause the video and like try these yourself if you haven't already, and you can go into the PowerPoint up and down. If you want to click on this link up here, it will take you to the actual page in the online file of the course description at the question if you want to like see the whole thing, because I copied pieces in places so you can see multiple pieces. So here is the answer to A. Okay. I also have a link up at the top if you want to see the scoring guidelines and everything as well. So what they're asking you in A is basically what causes amino acid changes or amino acid substitutions. We've changed a leucine to a phenylalanine, and you're not given any information about what was the DNA sequence or code, what RNA codons do we have present. You don't have a codon chart, so you don't have to get as technical into that. It can be literally as simple as a change occurred in the DNA sequence that encodes for that sodium channel protein. That's what it is in the rubric. I think you could say anything that there's a change in the DNA sequence or there's a change in a DNA nucleotide resulting in a change of one amino acid of this protein. I think that would still get to that describe point, and that's 1 point. Okay, and then explain how the substitution is resulting in resistance, right? So you're looking at, okay, you've changed one amino acid, how is this causing resistance? So now you've got to get into, normally when leucine is the amino acid, it's killing the mosquitoes, right? The insecticide is able to disrupt the function of this sodium channel pump, or sodium channel protein, right? And so it's killing those mosquitoes. But they say here phenylalanine is substituted and scientists hypothesize it's responsible for the resistance. So you have to connect the change in the monomer and how it has affected the polymer. So I've changed an amino acid, it's changed the shape or function of that protein, and now it has incurred further with a change in what is it called? Survivability of the organism. So their statement is the substitution change could change the shape of the protein channel so that the insecticides can no longer affect it, bind to it, or interfere with its function. You don't have to go into the properties of leucine or phenylalanine. You don't have that information, so you're not expected to like come up with it out of nowhere. That's beyond what you need to know. You don't have to identify how the protein has changed in shape, like that now this is outside of the membrane when before it was in the membrane. That's not a piece that you know. You basically just need to explain that this protein no longer interacts in a lethal way with the insecticide. It has now incurred resistance for the mosquito and the mosquito survives. So that is for A. Let's see how fast I can do B. Identify the dependent variable in the experiment whose data are graphed in figure 2. So if you go back and look at figure 2, we've got percent mortality and we have times, different times that they collected mosquitoes. You cannot change time. This was the time it occurred. This is the regions, right? And you are testing these two different insecticides. What you're recording as dependent upon those things is the percent mortality. So percent mortality or susceptibility to insecticide is fine too. They use that statement inside the prompt itself. If you were looking for an independent variable in this one, it would have been which insecticide are you using, which region are you in, what time of year, what point was the data collected. So the independent variable in this one, there's like a lot of field test like study data, but then what the only thing they're recording dependent upon that is did the mosquitoes die, what percent of them died. Identify the positive control in the experiment. So positive control is a way to confirm something indeed is happening the way it should, right? A positive control, like in the transformation experiment, is plating regular bacteria on a plate with antibiotics, but have not provided the, be negative. Well, they're dying. It didn't have the plasmid, but it does have the antibiotic there. So it would have like made the bacteria die. That confirms your antibiotic is working. So a positive control, I pulled this from the internet, a group or, bleh. Positive control in an experiment that receives a treatment with a known result and therefore should show a particular change in the experiment. It's confirming that what you are doing is working and that the pieces of your experiment are actually working the way that they're supposed to. So